Dear students, today we will discuss about genome mapping, which is part of our course, that is genomics and proteomics. The main highlights of our today's lecture is aim of genome mapping, importance. We will also discuss types of genome mapping, that is genetic mapping as well as physical mapping. While briefly we will discuss about the steps that is needed for making genetic maps. Genome mapping is important because it gives you information about the location of genes on the chromosomes. It also gives us information about the relative distance between the genes. In the era of genome sequencing, its importance is increasing because you know that for genome sequencing, fragmentation is the utmost step, and by fragmentation of the genome, the arrangement of the fragments is disturbed. So after sequencing, we have to assemble those fragments, and their orientation is disturbed. So if we do not know the genetic maps, we cannot able to assemble those fragments. So the assembling of the fragment is only possible if we know genetic maps, and genetics map give us the recognizable features, and by using those recognizable features we can able to assemble the whole genome. As we discussed earlier, the importance of genome mapping in the context of genome sequencing, it gives us information about the whole genome. So to reconstruct the whole genome after fragmentation and sequencing is only possible if we know genome maps, and genome map give us information about the recognizable features, and by using those recognizable features we can able to arrange the fragments of the genome. It also gives us the detailed picture about the genome organization as well. We have two main types of genome mapping. The first one is known as genetic map, or you can say genetic mapping or linkage mapping, while the other one is known as physical mapping. Genetic mapping is important and it relies on the relationship of the linkage, or you can say arrangement of genes is possible in genetic mapping on the basis of the recombination frequencies, and by using those recombination frequencies the relative distance between genes can also be find out, while physical mapping gives us information about the actual distance between the genes. We have variety of methods like non-PCR based method as well as PCR based method, which can give us the actual distance between the genes. For PCR based method we have rapid, AFLP, SSRs and SNPs. Besides this we have other markers as well, and these are the major markers by which we can find the actual distance. As we discussed earlier, that genetic mapping relies on the relationship of the linkage, or you can say relies on the frequency of recombination. And how we can determine the recombination frequencies? Recombination frequencies normally give us information about the linkage, which is described in the form of map units, and the relative distance between the genes is described in the form of map units, which is actually 1 centimorgan. 1 map unit is equal to 1 centimorgan, which explains the recombination, percentage of recombination. Recombination frequencies were earlier discussed that it relies genetic mapping relies on the recombination frequencies, and the recombination frequencies give us information about the relative distance between the genes in the form of map units. And it also gives us information about the level of linkage. Either the gene is linked or unlinked. If the relative distance between the gene is 50 centimorgan units or less than 50 centimorgan units, they are normally considered as linked genes. While those genes which have relative distance more than 50 centimorgan units, they are considered as unlinked genes. They are normally located on non-homologous chromosomes, and they do not follow under law of independent assortment. You know, law of independent assortment explains that if the genes are unlinked, they sorted independently. So means one gene have no effect on the other, while linked genes do not follow law of independent assortment. So genetic mapping can also be determined by using recombinant types. If the recombinant type is more, so mean recombination is more. If recombinant type is less, that means recombination is less. How we can determine the recombinant? So you know that during meiosis, chromosomes come together and form synapse. During synapse formation, or you can say the point at which they form synapse, that is called as chiasmata, they exchange chromatids, and exchange of chromatids is known as crossing over, and the crossing over basically give recombinations. Crossing over is not detected because no recombination between genes, and if crossing over occurs, recombination occurs. We have around 200 crosses in which crossing over occurs. One single crossing over occurs, and as a result of that single crossing over, we can get two recombinants, while 198 progenies are non-recombinants. By dividing the recombinant with the whole progeny, we can get the recombination frequency, which is described in the form of map unit, and map unit actually give us information about the centimorgan distance, relative distance. For making genetic maps, we have some criteria. The utmost criteria is contrasting phenotypes. Parents with contrasting phenotype is utmost important to making genetic maps. As you know that Mendel have contrasting phenotypes like tall pea plant and tall or dwarf pea plants. By crossing those, the first generation is called as F1 generation, or you can say first filial generation, and by the crossing of the first filial generation, we can get the second filial generation by and we can And after getting F2 progeny, there's need to get information, or you can say find the recombination frequencies. By those, nowadays we have a lot of softwares which can give us the detailed picture of the genetic maps by using those recombination frequencies. With this, we are concluding our today's lecture about genome mapping. The main material which is needed for us for detailed study, that is, we are getting from concepts and techniques in genomics and proteomics. Thanks for your attention.